So now I am back with my next video, and in this video we are going to talk about electron transport chain. So electron transport chain it happens after the Krebs cycle. So the NADH which are released during the glycolysis during the TCA cycle, they all fed into the electron transport chain to release energy and to synthesize ATP. So please make sure to like, share and subscribe my channel. So, what is an electron transport chain? It is a series of complex that transfers the electron from the donor to the acceptor via different reaction, where different redox reaction, and during this they transfer protons across the membrane. So the energy that is released during this transfer, they use it to transfer the protons across the membrane. So it is basically the transfer of the electrons from the NADH and FADH2, which is released in the during the Krebs cycle, the TCA cycle, to oxygen via different complexes, different multiple carriers. And also it is the process, as I have talked about, like the NADH FADH2, which is released during glycolysis, beta oxidation, other processes, and then thus releasing energy in the form of ATP. And the electron derive it is used to derive the synthesis of ATP from the from ADP. So the thing is, where is it happens? So it happens in the inner mitochondrial membrane, near the place where the TCA cycle happens. And the protons that are transported from the matrix, from the matrix of the mitochondria to the intermembrane space. So here you can see the mitochondrial matrix, the inner mitochondrial membrane, and the intermembrane space. So during the electron transport chain, the protons are transported from this matrix to the across the inner mitochondrial membrane to the intermembrane space. And then this process, as you can see, take place in a units arranged in the cristae of the membrane of mitochondria. So these are the complex that are located around the mitochondria, the cristae of the mitochondria, where this reaction happens. So there are different components of the electron transport chain, or you can say different complexes which are involved. So there are in total 4 complexes which comprise the electron transport chain, and each of the complex is composed of multiple polypeptide chains, or up to 40, and then they perform several functions such as enzymatic activities and transmembrane pumps as well. So there are different complex, as I have talked about. So the first complex is the complex 1, or the NADH dehydrogenase, because they dehydrogenate the NADH and uses this to transfer the protons use this to transfer the protons from the matrix to the intermembrane space. The second complex is the complex 2, succinate dehydrogenase, which uses FADH2 and transfer the electrons from FADH2 to the another complex. And the third one is the cytochrome BC1 complex, which is the intermediate between the transport to the oxygen. So it composed of different cytochromes which help in the transfer of the electrons. And then the complex 4, which is the last complex, in which the O2 they get the protons and releases H2O. So we'll talk about in detail about each of the complex. So the complex 1, so it catalyzes the oxidation of NADH with the reduction of coenzyme Q. So I will talk about what is coenzyme Q, and it is the largest protein in the mitochondrial membrane, containing 43 polypeptide and a molecular weight of approximately 850 kilodaltons. And it contains different iron clusters and FMN group as the prosthetic group, and it pumps 4 protons across the mitochondrial membrane. So here you can see the NADH, when it get oxidized to the NAD plus and releases H plus, the protons. And then during this process it releases electrons as well. And then these electrons moves from the FMN to iron cluster to the ubiquinol, this ubiquinone, which is also known as coenzyme Q, and produces, when two electrons, it produces QH2. and the energy that is generated during this process is used to pump four hydrogen ions into the intermembrane space. So one NADH during the complex one uh, transfers the pumps four protons across the intermembrane space. So what is coenzyme Q? It is a highly hydrophobic lipid soluble benzoquinone, and it accepts electron to become semi-ubiquinone, or it can, like here you can see the ubiquinone which is fully oxidized. It can accept one electron and form the semi-quinone radical, and then it can accept the another electron as well to form the ubiquinol. So this is called the coenzyme Q. So because it contains both electrons and protons, so it plays a role in the electron flow from one complex to the another. So because if there are different complex located at different position, it's difficult to transfer the electron. So coenzyme Q, it helps in the coupling of electron flow to the proton movement, and then it can help in the transfer of electrons to different complexes as well. So next we will talk about the second complex, succinate dehydrogenase. It contains four different proteins, and here you can see that this is the complex one we have talked about. So this is the complex 2. It has a protein which has the FAD prosthetic group, right? Flavin adenine dinucleotide, which transfers the electron from the FADH to FAD and releases past it to the FES complex, and then the electrons are transferred to the ubiquinol like the complex one, right? And then there are different electrons like the FAD group which can transfer, like the beta oxidation, the dehydrogenase, it transfer the electron to FAD, right? And then it moves to ubiquinol, or different glycerol 3 phosphate, they can also transfer the electron to FAD and then it passes to quinol. So all these different complexes, it can pass the electrons to the ubiquinol. And the important thing is, during this complex 2, there is no pumping of protons across the inner membrane space. So now is the complex 3, which is a dimer of two identical protein and each contain the 11 different subunits. So the major core is the cytochrome B, which contain the ferric iron cluster, ferrous iron cluster, and then the cytochrome C. And the electrons from the quinol, from the one where the complex 1 and complex 2 passed, they all get transferred via the iron cluster to the cytochrome C. So here you can see that they transfer the electron to the iron cluster, this ubiquinol, and then it passes to the cytochrome C. And during this process, the four protons are pumped across the mitochondrial membrane. So next is the complex 4. So it get like, now we have the electrons in the cytochrome C, 2 from each of the complex, and then during this process it transfers the electron to the oxygen and to form the H2O. So per H2O form there are 2 proton produced. So 4H plus produces 2 H2O if there is 2. So per formation of water molecule there are 2 hydrogen ions which are pumped. And then as I have talked about, like 2 protons are pumped to form the water, and then the protons which are formed in the inner mitochondrial space. So next is the formation of ATP. So complex 4 is the end of the electron transport chain, and then there comes another complex which is the ATP synthase, which is to produce ATP. So now because we have so many hydrogen, like the gradient is formed of the more protons in the inner membrane, like the intermembrane space, and then less protons in the inner mitochondrial membrane. So now there is a transport of the hydrogen ions from the protons from the inner membrane space, the intermembrane space, to the mitochondrial matrix by a complex which is known as ATP synthase. So ATP synthase, it consists of two subunits, F0 and F1, which are linked by a slender stalk. So F0 is a electrically driven motor, and it spans the lipid bilayer through which the protons enter the membrane. And then is the F1 complex, F1 subunit, which harvests the energy from this hydrogen proton movement, like the proton movement down the electrical gradient, to catalyze the formation of ATP. So when the energy that is produced, like the electrochemical gradient, like, so the ADP get phosphorylated to form ATP in the F0 complex. So this is the electron transport chain in summary. First complex is the dehydrogenation of the NADH, and during this process 4 protons are pumped. And then the complex 2, which is the succinate dehydrogenase, it transfers the electron to the coenzyme Q or to the quinol, but it does not pump any protons. And then the third complex, cytochrome BC1 complex, which pumps 4 protons. And then the fourth complex, which, like, per H2O, it releases two protons, and then these protons moves to form the ATP. So as you see, there are 4 protons which are required to produce one ATP. So in summary, in glycolysis we get 2 NADH and 2 ATP. With the Krebs cycle we get 8 NADH, 2 FADH2 and 2 ATP or GTP. So in total what we get is 10 from NADH, 2 FADH2 and 4 ATP. So if we see, like, we have 10 hydrogen which are pumped from the 1 NADH, and 4 are used for 1 ATP production. So in total 1 NADH can produce 2.5 ATP. So if we have 10 NADH, so from glycolysis to TCA cycle we produce 25 ATP from NADH, and similarly the FADH. So if we don't have the complex 1, for FADH, the protons are pumped only from complex 3, 4 protons, and 2 protons from complex 4. So we get 1.5 ATP from FADH2. So if we have 2, so we get 3 total ATP from glycolysis and TCA cycle, and 4 ATP we get. So in total, if we have the glycolysis and TCA cycle, we get in total 32 ATPs, which is why the TCA cycle is preferred over glycolysis for the source of energy. So I hope it's clear. If you have any doubts, please let me know in the comment section, and please like, share and subscribe my channel. Thank you.